Fruits like banana, orange, mango, and apples are plucked from the plant when they are unripe and are then allowed to ripe. What's the difference between a unripe and a ripe fruit? If we take banana as an example, compared to an unripe banana, a ripe banana is softer, loses green color becoming yellow, has a different aroma and sweeter in taste. All these changes happen after the fruit is detached from the parent plant. Biochemically speaking, the ripe banana has more sugars, less starch, the green chlorophyll is degraded, yellow carotenoids are synthesized, and the rigid cell walls are broken to soften the tissue. These biochemical changes are triggered in the unripe fruit by ethylene. This compound, ethylene, is the only plant hormone that is gaseous in nature and can diffuse. Although the biochemistry was understood later, because of the gaseous nature of the hormone, farmers knew that one ripe fruit can induce ripening in others. Manipulation of ethylene biosynthesis or inhibition is commercially used now to regulate ripening of the fruits. The importance of ethylene as a plant hormone was realized after the discovery of gas chromatographs, which could be used for quantification of ethylene. Ethylene regulates several physiological processes in plants like senescence and abscission of leaves, flowers and fruits, thickening of the underground parts of a seedling, growth of underwater parts of an aquatic plant, and epinasty or drooping of the leaves. Let us see in detail about ethylene. In earlier times, with no electricity, coal gas was used for illuminating street light. It was a common observation that trees near to these lamps shed their leaves more extensively than others. Much later, it was identified that ethylene is a component of coal gas. The Russian scientist Dmitry Nelyubov was the first person to study the physiological responses of seedlings to ethylene. He was also using coal gas lamp in the laboratory and found that dark grown pea seedlings in the laboratory had reduced stem elongation, increased thickness and horizontal growth habit compared to the seedlings grown in fresh air outside the laboratory. This response is now known as triple response and can be observed experimentally in all the seedlings exposed to ethylene. First observation on the ripening of the fruit was done by Cousins in 1910. He found that when oranges were separated in a chamber from unripe bananas, some gaseous emanations caused premature ripening of bananas. Ethylene was chemically identified as a natural product of plant metabolism in 1934 by Gane and was classified as a plant hormone because of its dramatic effects. Ethylene is a simple gaseous compound and is lighter than air. It is flammable and gets oxidized to ethylene oxide. In most plant tissues, ethylene is completely oxidized to carbon dioxide. All parts of the plants produce ethylene, but the production is highest in senescing tissues and ripening fruits. Methionine is the precursor of ethylene in plant tissues. It is first converted to S-adenosyl methionine. In the next step, the immediate precursor of ethylene, aminocyclopropane carboxylic acid or ACC, is synthesized. This reaction is catalyzed by the enzyme ACC synthase. ACC is further oxidized by ACC oxidase to form ethylene, carbon dioxide and ammonium. The methionine is recycled via the Young cycle. The control of production of ethylene is regulated by the activity of ACC synthase and ACC oxidase enzymes. The activities of these enzymes are enhanced in the ripening fruits. Inhibition of the activity of these enzymes can keep the fruits in an unripe condition. Chemicals like aminoxyacetic acid and aminoethoxyvinyl glycine can inhibit ACC synthase enzyme. ACC oxidase enzyme is inhibited in the absence of oxygen, in other words, under anaerobic conditions. Commercially, inhibition of these enzymes is used as a method to keep the fruits in an unripe condition for storage and transport. Exposure to ethylene changes the growth pattern of most of the dicot seedlings, like in pea, and causes triple effect. In these seedlings, ethylene inhibits stem elongation, increases stem thickening, and causes horizontal growth habit. Thickening has a survival value for the emergence of seedlings from the soil. When seeds germinate below the soil surface, they have to initially push through the soil. The resistance of the soil will be higher than the resistance of air. Thickening helps to overcome this resistance. Once they emerge from the soil, further growth will happen in less resistant air. In Arabidopsis, the triple effect consists of inhibition and swelling of hypocotyl, inhibition of root elongation, and exaggeration of the apical hook. All seeds require water in the soil for germination, but excess water inhibits their growth. Only aquatic plants can grow underwater, whereas the rest can grow only in moist soil. In aquatic plants, ethylene is able to promote the growth of stem and leaf petioles underwater. The important crop rice is an aquatic plant that grows in standing water. In these plants, stem grows faster when they are submerged. Treatment with ethylene when they are not submerged mimics the effect of submergence. Under submergence, ethylene builds up in the tissues as loss of ethylene due to diffusion is retarded underwater. Ethylene decreases the level of the growth inhibitor abscisic acid and enhances the sensitivity of the tissue to the growth promoter gibberellin. Lotus, Ranunculus scleratus and Nymphoides peltata are the other aquatic plants in which growth is enhanced in the submerged parts by ethylene. Root growth. Ethylene is a positive regulator of root hair formation in several species. Root hair formation is promoted by ethylene in seedlings of lettuce and Arabidopsis. Root hair formation is reduced in presence of inhibitors of ethylene. Ethylene induces flowering in plants like pineapple and mango. In pineapple, ethylene is commercially used to synchronize fruit setting. In monoecious species, that is, plants that have separate male and female flowers, ethylene may change the sex of flowers. In cucumber, for example, ethylene promotes formation of female flower. Epinasty is the downward curvature of the leaves. This curvature happens when the upper side or adaxial side of the petiole grows faster than the lower side or abaxial side of the petiole. Ethylene and Oxen induce Epinasty. Oxen causes Epinasty indirectly by inducing ethylene production. Flooding of plants like tomato causes epinastic response of the plants. The waterlogging is sensed by the roots, and since oxygen diffuses slowly through water, it creates an anaerobic condition around the roots. Decrease in oxygen prevents oxidation of ACC to ethylene in roots, and ACC accumulates. The ACC is then transported to shoots via xylem, where they are readily oxidized to produce ethylene and cause epinasty. In this manner, ACC acts as a signal from roots to shoots under waterlogging conditions. Exogenous ethylene accelerates leaf senescence. Enhanced ethylene production is associated with chlorophyll loss and color fading. Inhibitors of ethylene synthesis can retard leaf senescence. Senescence is a genetically programmed developmental process, and it is regulated by abscisic acid, ethylene and cytokinins. Abscisic acid and ethylene promote senescence, and cytokinins retard senescence. Abscission or shedding of mature leaves, flowers and fruits takes place in a specific layer of cells called abscission zone. In this zone, cell walls of the abscission layer are weakened by the wall degrading enzymes like cellulase and polygalacturonase. These enzymes are promoted by ethylene, which acts as a primary regulator of abscission. In the abscission zone, oxen concentration becomes lower and ethylene production is increased. When a fruit is ready to eat, it is a ripened fruit. Otherwise, it is a raw fruit. A raw papaya is green and not sweet, but a ripened papaya is yellow and sweet. Fruits change their color when they ripen. They degrade green chlorophyll and synthesize yellow carotenoids or red anthocyanins. A papaya fruit turns from green to yellow, but a tomato fruit turns from green to red. Besides color change, the fruits soften while ripening and degrade starch to sugar like in banana, or become sweet from sour taste like in orange by converting organic acids to sugars. The color change in fruits attracts animals to edible fruits by making them more visible, and it is an adaptation to ensure seed dispersal. A green colored raw fruit merges with the green color of leaves and is not readily visible. Birds and other animals disperse the seeds of edible fruits to a large area. The ripening of the fruit is regulated by ethylene because there is a rise in ethylene production before ripening. Exposure of raw fruits to ethylene hastens the process of ripening. Fruits like banana, mango, tomato, apple and fig respond to external application of ethylene, and these fruits are called climacteric fruits. But there are some fruits like pineapple, grape and strawberry which do not respond to external application of ethylene, and they are referred to as non-climacteric fruits. In climacteric fruit ripening, ripening can be delayed or inhibited by the application of chemicals like AOA, that is aminoxyacetic acid, or AVG, that is aminoethoxyvinyl glycine, which can inhibit the biosynthesis of ethylene. Elimination of ethylene biosynthesis in tomato by silencing the genes that code for ACC synthase and ACC oxidase enzymes, which are crucial to ethylene biosynthesis, completely blocks the fruit ripening. Genetic studies in tomato have revealed that ethylene regulates the expression of genes involved in the synthesis of lycopene, a carotenoid, aroma biosynthesis, ACC synthase enzyme, and respiratory metabolism. Other genes which code for chlorophyllase, responsible for degrading chlorophyll, ACC oxidase that converts ACC to ethylene, and polygalacturonase responsible for cell wall hydrolysis are also expressed while ripening as a part of developmental process. However, they are independent of ethylene. In general, external application of ethylene is used for hastening fruit ripening, and the inhibitors of ethylene biosynthesis are used for fruit preservation, transport, and enhancement of shelf life. Ethylene is commercially applied as a liquid spray in the form of ethefone, also known as ethyl. This compound is 2-chloroethyl phosphonic acid. This is absorbed and transported in the liquid form by the plant, and it releases ethylene slowly by chemical reaction when it breaks down to chlorine, ethylene and phosphonic acid. Ethaphon is used for hastening ripening in tomato and apple. Other uses of ethaphon are synchronization of flowering and fruiting in pineapple, fruit thinning and fruit drop in cotton, cherry and walnut, promotion of female sex expression in cucumber flowers. In order to reduce the rate of ripening and increase the storage period, fruits are stored in low temperature and low concentration of oxygen that prevents production of ethylene. A high concentration of carbon dioxide, that is 3 to 5%, also prevents ethylene action as a ripening promoter. Inhibitors of ethylene action like silver ions applied as silver nitrate or as silver thiosulfate are used to increase the longevity of cut flowers like carnations. In future, many agricultural plants may be genetically modified to regulate ethylene biosynthesis and control the ripening of fruits and retain freshness of flowers. Ethylene is the only gaseous plant hormone that regulates several developmental processes in plants: emergence of seedlings from the soil, growth of underwater parts of a plant, fruit ripening, and abscission of leaves, flowers and fruits are the important processes regulated by ethylene. Since manipulation of ethylene biosynthesis or ethylene action can regulate fruit ripening and retention of freshness in cut flowers, ethylene has commercial importance.